All right, that looks fine, eh? Yeah. Okay, so great to be here. It's always good to get out of Canada and come to California, especially when it's close to winter. All right, so the main event in the red corner, hailing from an alpha proteobacterium, we have the sprightly mitochondrial DNA, and in the blue corner, born and raised in a cyanobacterium full of the sun's energy, we have plastid DNA. And based on the talk so far, I think mitochondrial DNA is winning. So anyway, today I want to talk about the evolution and origins of genome architecture within mitochondria and plastids. And specifically, I want to compare their similarities, their differences, unique features, the eccentricities found in each one of those compartments. And ultimately, what I'm going to do is I'm going to try to convince you that mitochondria harbor a greater breadth of genomic complexity than plastids. So I'm going to say that mitochondrial genomes are generally more extreme, they have more pronounced eccentricities, and more severe embellishments than plastid genomes. And then I'm going to finish with some discussion of why this may be. Okay. So there's mountains and mountains of data on organelle genomes. In fact, they're still the most sequenced type of chromosome, more than viral genomes, bacterial, and obviously more than nuclear genomes. So as of the end of the summer, there's 5,000 complete mitochondrial and plastid DNAs available. And if you break those down, it looks something like this. So most of those sequences come from mitochondrial DNAs, about 90%, and only about 10% come from plastids. And you're probably not surprised if you look at those mitochondrial DNAs, most of them come from animals. So whatever you do after this talk, for Christ's sake, don't go out and sequence an animal mitochondrial genome. All right, so the interesting stuff is found in here. This is where most of the eukaryotic diversity is found. And if we break this down further, it looks something like this. So here, these two dark green slices, they represent the mitochondrial and plastid genomes from plastid-bearing protists. And here's land plants there. So I guess what I'm getting at is that although there's an order of magnitude more mitochondrial genome sequences than plastids, plastid genomes are actually very well sampled when you think of eukaryotic diversity. In fact, for many lineages, including green algae, red algae, diatoms, many alveolates, we actually have a better understanding of plastid genome diversity than mitochondrial genome diversity. So ultimately, given the available data, I think we can make kind of meaningful and broad level comparisons between those two types of genomes. And what have we learned from all this data that's out there, right? All this organelle genome data. So, it's been wonderful for understanding eukaryotic evolution, reconstructing trees, for forensics, anthropology, archaeology, for industry, for transforming those genomes. But kind of more than anything, these data, they've been kind of an endless reservoir for kind of bizarre and unconventional genomes, genomes that break all the rules and are just kind of really cool. And that's true. Like mitochondrial and plastid DNAs, they're funky on so many levels, right? They can have weird compositions, structures, their sizes, the way they're expressed, the types of foreign DNA found in them. And you could spend a whole course or write a whole textbook just on this one topic. And obviously I can't go and describe all of this. But what I want to do for the next kind of half dozen slides is kind of highlight some of the more extreme organelle genome architectures that are out there. So, as you're probably all well aware, there's kind of a near universal adenine and thymine bias in organelle DNA across the eukaryotic tree and across endosymbionts in general. But GC-rich mitochondrial and plastid DNAs, although rare, they do exist. So the highest GC content that's really ever been recorded in an organelle comes from the mitochondrial DNA of this seedless vascular plant, Selaginella. And its GC content is significantly higher than the record for plastids, which comes from this Trabuxiophyte down here. And interestingly enough, this is actually super cool. All species with GC-rich plastid DNA also have GC-rich mitochondrial DNA. They always occur together, but the reverse is not true. You can find GC-rich mitochondrial genomes alongside AT-rich plastid genomes. So we often envision organelle chromosomes as genome-sized circular mapping structures, but they can be much more multifarious. So linear mapping mitochondrial genomes with defined telomeres have evolved no fewer than 20 times across the eukaryotic tree and in diverse lineages. And sometimes these telomeres can be very elaborate with closed loops, overhangs, covalently bound proteins. But conversely, linear mapping plastid genomes have really just been described from one species, and that's Chromera, which is a photosynthetic close relative of AP complexans. And many, many times in mitochondrial evolution, an intact chromosome has been fragmented into multiple pieces, and that can involve linear or circular type chromosomes, and the levels of fragmentation, they can be quite minor, like in this non-photosynthetic green alga or this rotifer, or they can be moderate, like in this head louse or in jellyfish, or they can be extremely severe, right? As we've heard in some of the earlier talks, like in Ammobidium or Diplanema. So anyway, if you go out swimming over the next day or two and you get stung by a jellyfish, console yourself by how cool its mitochondrial genome is. Or maybe you'll get a bit of lice from the hotel, hopefully not. But again, now on the other hand, really fragmented plastid genomes are confined to just a single lineage. Dinoflagellates, particularly the dinoflagellates with peridinin-containing plastids, like this Symbiodinium right here. And we'll hear more about that, I think, in some of the coming talks, with these remarkable mini-circular plastid DNAs, which really are funky and very similar to the lice mitochondrial genomes. So the biggest organelle chromosomes we've ever observed are found in the mitochondria of seed plants, like Silene canonica here, which we heard about earlier today. 11,000 letters, 11 million letters long, only 1% coding. And these mitochondrial genomes are significantly bigger than the biggest plastid genomes we've ever observed, from things like Volvox, about 500,000 nucleotides, and this Ulvophyte acetabularia down here, which is maybe 1.5 million. And at the other end of the extreme, we have these highly, highly reduced mitochondrial genomes in AP complex and parasites, just 6,000 kilobases and only 5 genes. And Chromera may even have a smaller mitochondrial genome, just a few thousand nucleotides, even less. And certainly plastid genomes can be small as well, right? But like in Plasmodium and other non-photosynthetic plastid-bearing protists, they can be around 30 kb, but they're always quite a bit larger than the smallest mitochondrial genomes. And many times in mitochondrial evolution, there's been the evolution of a non-standard genetic code. In animals alone, you have 12 different changes to the genetic code with up to 5 codon reassignments in a single species. And most protists have undergone 1 or 2 changes to their mitochondrial code. Whereas in plastids, particularly in primary plastids, you don't find any significant changes to the genetic code. And there's mostly just a few unconfirmed reports of changes to the genetic code in other types of plastids, like in this dinoflagellate down here, which John mentioned earlier, which has a green algal plastid, has a single change to the code. And RNA editing has appeared in all kinds of different mitochondria, from slime molds, land plants, dinos, for example. The most intense type of editing is likely in trypanosomes, where 90% of the codons undergo insertion deletion editing. And plastids too can experience RNA editing. In this hornwort down here, there's about 500 CDU and another 500 U to C editing sites. But overall, the levels of RNA editing that you find in plastids are not nearly as as significant and as you find in mitochondria overall. So I could keep going and going, giving you examples of extreme organelle genomes. It took me a long time to do this figure. I just wanted to have a good old look there. But I'll stop, because I think you know what I'm getting at. So in many ways, organelle genome complexity within mitochondria mirrors that of plastids. Both have independently adopted kind of almost identical architectures and similar embellishments time and time again, right? But in mitochondria, the intensity of these embellishments is generally much more pronounced than it is in plastids, and much more widespread, okay? And in fact, you can see these patterns, patterns like this within a single cell. So Selaginella, which I mentioned earlier, it's an excellent illustration of how both the mitochondrial and chloroplast genomes can adopt almost parallel architectures within a single organism. So both genomes have some of the highest GC contents we've ever observed, record levels of cDNA editing, a reduced tRNA content, they're intron-rich, and the structures are quite rearranged compared to close relatives. But for all these traits, you can see that the mitochondrial genome outdoes the plastid. It has a higher GC content, a greater number and fraction of editing sites, fewer tRNAs, more introns, and generally it's much more structurally rearranged than the plastid. So then the question becomes, why? Why do these two types of genomes, why do they often take very, very similar paths, sometimes in very different lineages or sometimes in the same cell, but why does the mitochondrial genome always get pushed further in most instances? So we could argue that maybe mitochondrial DNAs are just better sampled than plastid DNAs, and we haven't really observed the full breadth of complexity in plastids yet. But I think I've argued well at the beginning by saying that's not true. We have a very good understanding of plastid genome architecture. You could say, well, mitochondria came first. They've had more time to get extreme than plastids, right? But I don't like that either, because in many cases, these extreme embellishments that you find, like RNA editing, they arose almost at the same time in both plastids and mitochondria, for example, in land plants, or the expansions that you find in some lineages. They've happened relatively recently. Certainly, there's some physiological and structural differences between mitochondria and plastids that can account for differences in their genome architecture. So, for example, unlike plastids, mitochondria regularly fuse in vivo, and we know that they, at least in some lineages, there's an active DNA import system, which we haven't well defined in plastids. And this can explain, for example, why you'd find some organisms with all kinds of foreign DNA in their mitochondrial genome, like the shrub here, Amborella, which actually has the equivalent of 6 foreign mitochondrial genomes within its own mitochondrial genome. It's remarkable. And why you don't find something like this with plastids. But really, that's just accounting for a very small aspect of genome architecture, just foreign DNA. I think to understand kind of why mitochondria harbor a greater breadth of complexity than plastids, you really have to kind of get into the underlying forces that fashion chromosomes, so namely mutation, recombination, selection, genetic drift. So there's increasing evidence that non-adaptive processes, as opposed to selective ones, have a huge impact in shaping organelle genome complexity and architecture. So, for example, something like constructive neutral evolution, which is a non-adaptive ratchet-like process. This can eloquently explain the gratuitous levels of RNA editing that you find in something like kinetoplasts. But we're still left trying to understand why mitochondria may be more prone to these mutational ratchets than plastids, right? So Michael Lynch put forward a universal non-adaptive hypothesis to explain genome architecture and evolution across the tree of life, and he focused a lot on organelle genome evolution, and arguing that kind of genome embellishments, so things like introns and RNA editing sites and repeats and all these types of things have a greater tendency to accumulate in lineages with very low mutation rates and small effective population sizes with lots of random genetic drift. And surely you see that some of these very, very embellished organelle genomes, such as the Volvox plastid genome, which again is very bloated, or some of these land plant mitochondrial genomes like that of the tulip tree, which are very inflated, they have very, very low measurable levels of NEMU, which supports the hypothesis. But to be fair, when you start exploring more lineages, the trend starts to break down slightly. So just one example would be silene, which we mentioned earlier, which surely has one of the most embellished mitochondrial genomes out there. It's very, very large, but it has a very, very high measurable mutation rates and no obvious reductions in effective population size. So again, not supporting this idea that NEMU is the main component driving organelle genome diversity. But one thing that has come from these studies is that extreme organelle genome architectures are often associated with very extreme mutation rates, be they high or low. Now, in many ways, mutation rate is a reflection of the underlying DNA maintenance machinery that are found within organisms. So this, in some ways, this could suggest that organelle genome architecture, in some ways, it may be very, very strongly connected to the underlying DNA maintenance machinery within organisms. And actually, this is exactly what Alan Christensen found, particularly with respect to plant mitochondrial genomes. So he was looking at Arabidopsis mitochondrial DNA, and he described how within this system, there's likely two different types of double-stranded break repair that are going on, but they can actually explain some very interesting aspects of the mitochondrial genome architecture. It's very, very interesting. So within genes, it looks like there's a bias towards gene conversion, which is very accurate and gives you a very low observable mutation rate. Whereas within the non-coding regions, there appears to be a bias towards break-induced replication, which if you look at the way that works, it can lead to this expansion of non-coding regions and also the rearrangements among non-coding regions. So just by looking at repair and these two different types, you can explain kind of very two contradictory features of land plant mitochondrial genomes. One, they're incredibly low mutation rates when you look at genes, but two, they're massive levels of expansion within the non-coding regions, which is very interesting. So thinking of this, what one could say that maybe these underlying DNA maintenance processes are having much broader influences on organelle genome architecture across the eukaryotic tree, not just within land plants. So what we do know is that in plants and algae, many of the nuclear encoded mitochondrial targeted proteins that are involved in repair, recombination, replication, they have paralogs that are targeted to the plastid. And a large proportion are dual targeted, so they're targeted to both. And that can kind of, when you think of it like that may explain why you often find very, very similar organelle genome architectures within a single cell, right? Like bias towards GC in both. So I like that. But that being said, the evolution of these DNA maintenance processes are remarkably complex. They involve gene co-option, duplication, loss, lateral gene transfer. So it is a really hard thing to wrap your head around. But if what I'm saying is true, that they do have, they are impacting organelle genome architecture in a very strong way, and mitochondria, you agree with me, have kind of a broader complexity of genome architectures, then you may expect to find that these underlying DNA maintenance processes are much more finicky and capricious within mitochondrial systems than they are within plastids. And if this were true, you may expect to find very large differences in mitochondrial mutation rates across the tree of eukaryotes. And that's exactly what you find. So seriously, mitochondrial mutation rates are all over the place, right? They're some of the lowest rates of mutation we've ever observed in some organisms, like in land plants, and all the way to some of the highest mutation rates that have ever been observed. So this is cool. If you look at angiosperms alone, you can find a 5,000-fold difference in the rates of synonymous substitution. And you can also find a 350-fold difference in synonymous substitution rates within a single genome. So it's remarkable. And similar findings come from when you look at cnidarians, for example, and various other protist lineages with their mitochondria. So I guess what I'm saying is that the mitochondrial DNA mutation rate spectrum is very, very massive, and that's true both within lineages, among lineages, and sometimes within a single genome. And that based on what we know about the mutation rate spectrum within plastids, it's not nearly as broad. So again, kind of supporting this idea that the underlying DNA maintenance machinery may be much more variable for mitochondria than for plastids. So a lot of my work over the last 4 or 5 years has really been looking at the rates of evolution within an organism among the 3 different compartments. And I just put this up here. So you can't, these aren't absolute rates of evolution. This is synonymous site substitutions among the 3 genomes within a single lineage here. But the mitochondrial rates are in blue and the plastid ones are in green. And you can just see that when looking across some of these groups, it truly is very, very variable when you focus on the mitochondrial DNA. And one really interesting thing is that in dinos, which some would argue have the most complex plastid genomes, like in Symbiodinium, you do find this very high level of substitution in the plastid genome relative to the mitochondrial and nuclear. So what I like about the idea that a lot of the complexity of organelle genomes is involved with the DNA maintenance machineries is that it can kind of explain why when things go wonky, they go really, really wonky, right? It's not just one thing. Often when you see a bizarre organelle genome, it's bizarre on so many levels in terms of the structure, the organization of the genes, the expression, and so forth. So I think that in some ways that explains this a little bit, and I like that. Okay, so looking forward, so I think that as we go from 5,000 to 10,000 to 100,000, to an infinite number of organelle genome sequences, I think that these trends will hold. I'm willing to predict that you will consistently find that mitochondrial genomes have a broader level of complexity than plastid genomes. I'm going to stand by that. You can smack me across the head later if you want. It doesn't hold. And then the other thing is when looking at kind of major breakthroughs that have happened, just in reading the literature in terms of our understanding of organelle genome evolution, a lot of it has come from having sequences from very, very closely related species, either multiple isolates from the same species, from like different geographical isolates, or very, very closely related different lineages, allowing you to kind of understand the molecular evolution between those 2 things. So I think having lots of data from different things that are very closely related is really, really important for understanding some of the underlying forces driving genome evolution. Yeah, and then I'll end my relatively lighthearted talk with a relatively lighthearted slide, like this. And and I'll just thank a lot of these ideas that I've been kind of talking about. They came about with discussions I've had with Patrick when I was in his lab, and also particularly with a grad student at the time who was in the lab, Jan Janusevic, who we talked about this a lot with. So, and I'll thank you for your attention.